Hello, everyone. As you know, this is our seminar series. We're meeting on a weekly basis with very exciting research happening at J Clinic. And well, today we're going to have Professor Sixian Yu. I'm going to read a short bio. Sixian Yu is an assistant professor at the Electronic Engineering and Computer Science department at MIT. And she's the principal investigator of the Computational Biophotonics Lab at MIT RLE. Her research focuses on developing microscopy technologies for biomedical problems through the lens of optical physics, instrumentations, and algorithms. Dr. Yu earned her PhD on microscopy from the University of Illinois at Urbana-Champaign, and she has been the recipient of many awards, just to list some, like the Shylock Award on Advancing Bioimaging, the Amazon Research Award, and the Microscopy Innovation Award. So the way we're going to run the session today is we're not going to have a clinical collaborator. I think it's going to be more like a talk, right? And then if there's any interruptions, I think if you're comfortable, just please feel free to interrupt. We can always skip a few minutes for the end. Usually our group is pretty engaged and they tend to ask questions, but feel free to run the show. The topic today is optics and machine learning for real-time cancer diagnosis. And well, I think at this point we can start. Sixian, the floor is yours. Welcome to the seminar series.
Thank you so much for the kind introduction, Ignacio. Let me share my screen. Can you see my screen?
Not yet, but yeah, almost there. Yes, beautiful. Now, awesome. Very well. Thank you.
Yeah, so give me 1 second.
And it's on, yeah, on presentation mode. Yeah, that's good.
Yeah, all right. So thank you everyone for attending the seminar. My name is Sixian Yu. I joined MIT last year, so it has been a very exciting ride for me. I'm very happy to be here to have the opportunity today to talk about our recent work in real-time optical biopsy that combines optics and artificial intelligence to make point-procedure diagnosis possible. So in the beginning, I want to give a shout out to our team at UIUC, where most of this work that I'm going to present today was done. So it does take a huge team to develop a technology and translate to clinics. So I want to give a shout out to the optical scientists and electrical engineers and the computer scientists, especially the clinical collaborators we had in Champaign, Illinois. And at Boston, I very much look forward to developing such a, or even more dynamic team with all the power, engineering power and clinical power at Boston. So first, why do we care about optical biopsy and what is it? So optical biopsy is a standard way to assess tissue in both hospital settings and also in biology research. Usually we start with something called H&E and immunohistochemistry. So what it does is that we take a tissue that has already been removed from either animals or human patients, and then we fix them, we embed them in the wax, and then section them using knives or microtomes, and then stain them using different dyes. And by doing this, it creates this unique contrast and resolution that is indispensable for tissue assessment and diagnosis of all kinds of diseases. So compared to MRI or CT or PET, that more people are more familiar with, so microscopy is something that is more hidden away from the public view, because what happens with microscopy is that it happens when the tissue is already taken from the patients. So everything happens in the pathology room. As powerful as it is, there are several major limitations that prevent a higher impact or a even higher accuracy of this conventional histopathology. The first thing is that it is time consuming. As you can imagine, all these steps are very manual, and it does take time to fix the tissue, to put it in the formalin, and to embed this, and also to section this. So it cannot be done in real time. So if I want to assess a surgical specimen, by the time I already finished the assessment with histology, it's very likely that surgery is already done, and we might need a second surgery if the surgery was not successful. And then the second limitation is that it can be very labor intensive. You need to train technicians and pathologists for many years in order to reach a consistent diagnosis based on the same specimen. And then the third thing, which is the most important thing in my opinion, is that we are looking at dead samples. When it's removed from the tissue, from the human bodies or the animals, what you have is a dead tissue that is dissociated with from the tumor microenvironment where the sample has grown and prospered. So a lot of diseases mechanisms can be much better studied with in vivo imaging when the sample is still alive and is still in its authentic 3D context. So there is a huge opportunity for us to move pathology from a dead section tissue to a in vivo setting. And this challenge is the trade-off between how do we do this in living samples, but still maintaining the molecular contrast and the subcellular resolution that make microscopy so attractive in the beginning. One of the potential candidates is nonlinear optical microscopy. So this is what we have as a state of art for a histopathology, where we stain them and section them, which takes hours to days to arrive at this image. So if we use nonlinear optical microscopy, instead of manipulating the tissue with stains and sections, now we are engineering the light to leverage the nonlinear interaction between the light and the tissue to give us the intrinsic contrast that will be important for diagnosis. And by doing this, we don't need to process the tissue in any way. We basically just shine the laser onto the tissue and to get the stain-free microscope images within minutes. And then even better, with more light engineering to speed up the process, the real attractive thing here is that we don't have to look at samples again. So we can actually look at how cells are moving in the living bodies in real time. So this is a video captured from a rat with memory cancer. So we did not do any genetic mutation, or we did not do any staining with respect to the animal. So basically it's an animal injected with tumor cells, and then we are taking a look at inside the tumor microenvironment and directly use the intrinsic contrast from the cells and the vessels to look at what is going on here. So by using nonlinear optical microscopy, we have the potential of removing the steps of staining and also sectioning to directly image the cellular and molecular contrast within the tissue to make the diagnosis. So what is the role of AI in this if nonlinear optical microscopy already did these many wonderful things? So the difficult thing is that this technology already existed. There are a few hurdles to really translate it to the clinical world. First challenge is that it's label-free. So being label-free is both a blessing, but also a curse. The blessing is that you don't have to label anything, so it's very friendly for clinical translation. But the curse is that because we don't have labels, so if you give our images to pathologists and biologists, the first question is what we are looking at. What are we looking at? There is no labels. How do we know what is what? So the first thing we did is to use artificial intelligence neural network to translate between the gold standard existing technologies, which is the histopathology, the stained microscopy, with our label-free microscopy. And then on top of that, since now we can translate between the two, can we use what we have in the new technology to discover new contrast, to discover new biomarkers? Because the intrinsic molecular contrast can carry chemical contrast and also structural contrast that was not conveyed in the original modality. And the third thing is that since we can now do it in the operating room, can we do this in real time to achieve point procedure diagnosis? So these are the three projects I'd like to go into today to give you an overview how we combined optics and artificial intelligence to facilitate in vivo and real-time optical biopsies. So first, in vivo pathology. Before we dive into the projects, I learned that the audience is mostly familiar with artificial intelligence. So I want to give a very general and a brief introduction into nonlinear optical microscopy, including why it gives us contrast, even though we did not do any labeling. So the contrast all originated from the nonlinear interaction between light and matter. If we hit the tissue with a really strong femtosecond pulse, not as strong as to destroy it, but as strong as to elicitate nonlinear processes, based on the molecules, for example, the chemical content of molecules or the structural content of molecules, it can convert the excitation photons to new emission photons. And based on different properties, it can give you different intensity and different color, which is a result of a wide range of nonlinear processes. And we don't have to understand what each name means. We only need to know that different modality or different nonlinear processes give us different contrast. For example, this is a nonlinear optical microscopy of exercised human breast tissue. Everything is fresh. It's just taking out the human patient who went under lumpectomy. So using the microscope we have, shooting the femtosecond laser into the tissue, we can visualize the fluorescence from metabolic products using all the fluorescence modalities like 2-photon fluorescence and 3-photon fluorescence. And by doing harmonic generation, like second harmonic generation and third harmonic generation, we can look at the unique structures of different tissues. And the new colors that's generated here in these images are actually intrinsic colors from the tissue. So, and the different color generation is because the tissues, you can think of it as a crystal, is converting the incident photons to different colors because of they have different absorption or conversion properties. And by analyzing these colors and their intensities, we can reverse engineer what was in the tissue. And then we can analyze how these metabolic properties, how these structural properties can indicate cancer status. So to give an example of what this label-free microscopy is capable of, this is a mosaic image. Basically means that we scan a large area in the living rats with memory cancer. So you can see there is a lot of vessels. The blue things are the vessels running across the rat. And then we can also see the tumors that are clustered here and also invading everywhere in this tumor microenvironment. And as we zoom in, we can even see more details in the tumor cells and also these vascular endothelial cells. And they look different colors because different colors carry different metabolic signatures. So for example, if it looks more bluish, that means they rely more on glucose metabolism, which means that they tend to reject oxygen as the ATP production, versus if they look more orangish, that means they have more oxygen in their ATP production, which is highly related to how cancerous these cells are. So in addition to a huge mosaic imaging where we can see the landscape of the cancer imaging, we can also look in real time how cells are moving. So this is a video captured in the same animal, but with a time-lapse video. So you can see that these cells are moving around and they are escaping from the vasculature. So the flowing things, these streams are the vasculature, and then the cells are flowing in and out of the vasculature to form immune cell clusters to fight inflammation in the tumor microenvironment. So by doing this in vivo and without introduction of any labels, we have the capability of capturing cellular and molecular dynamics in living bodies. And then when we apply this to pathology, we can compare with conventional histology. So we have very comparable structures with even more metabolic and functional contrast from the intrinsic contrast. And then the difference is that the second row, which is from the conventional histology, takes hours to days to arrive at these results, versus the label-free histology is all real time. So the first challenge we mentioned that for translation this into clinical world is that it does take a few years to understand what nonlinear optic phenomena means. So a straightforward thing to do is to use neural network to translate the nonlinear optic phenomena directly to biological objects using segmentation network. So what we did here is to use a Valina UNet, but we did not get very promising results by just using UNet. And then when we were looking into literature, there has been tons of work of doing multimodal MRI for segmentation. And it appears to us that if we do this with different modalities first, so basically instead of fusing the modalities in the low level in the beginning, but instead of that, we can process individual images in individual channels and then fuse these higher level features later, which actually gives us better results. And then we realize this is because our modalities, unlike RGB channels, which are usually processed by UNet, our multimodalities are actually pretty orthogonal to each other. So they do provide very complementary information, which can be actually more helpful if we analyze them first individually and then combine them together later. So that concludes the first project that combines optics and artificial intelligence to do in vivo pathology. How do we do the virtual staining? So instead of labeling with real dyes, how do we use intrinsic optical contrast and also neural network to do virtual staining? The second project is a natural extension of the first project, is how to use this technology to find new biomarkers. So here we have the capability to segment all the biological entities, and so we can quantify them and overlap the segmentation mask with the quantitative measurements with the segmentation mask to arrive at the quantitative analysis. For example, how many tumor cells are here and how many of them are metabolically active to do this kind of analysis. So in addition to the tumor cells, we are super interested in this new player in cancer development. It's called extracellular vesicles. So this was not really in the hot topic until very recently because these extracellular vesicles are super tiny. They are usually between 20 nanometers to 2000 nanometers, which is usually beyond the resolution of optical microscopes. And because they're so tiny, so they carry very little cargo in them, which also renders them very low contrast when you look at them using TEM or other imaging systems. However, because of the recent technology in transmission electron microscopy or SEM, people have found that this is actually the driving force of carcinogenesis. So because tumors do not have the mobility they want, so they actually send out these little soldiers, these little dots to different places in your body to either prime the environment so that the metastasis will be easier later, or to send soldiers to coordinate with their neighbors, future neighbors, to create a future site for the primary tumor cells. So, and this label-free microscopy comes in because it has bright signals for these extracellular vesicles because it has very strong signals due to the conversion in the third harmonic generation and also 3 photon fluorescence. Because of this, So we can visualize these extracellular vesicles and segment them using neural network, and then overlap the segmentation mask with the original intensity mask to get the metabolic analysis of individual extracellular vesicles. And we applied this to human breast cancer. These are all unprocessed tissues from the surgical specimens. And we found that when we do individual analysis of the extracellular vesicles, the cancer-associated extracellular vesicles carry surprisingly high NADPH, which is a reflection of the antioxidant defense mechanism. And that could provide us some insight on why there is always a trend of antioxidant metabolic activities in the cancer tissues. And then the second question we have in terms of new biomarkers is that would they have clinical utility? So stage 1 breast cancer is very hard to differentiate from other breast diseases. So there has been a lot of research on how to improve this. We thought extracellular vesicles could be an angle to solve this problem because this is a precursor to the cancer. And when we did analyze this metabolic analysis of these individual EVs, we found there is a high correlation between the NADH EVs and the human breast cancer diagnosis. So we're very looking forward to moving this forward for more clinical verification. And then the third project I want to introduce is tumor detection. So we talked about how we use the new optical technology and also the neural network for virtual staining to find new biomarkers. And then the third project is about how do we leverage the real-time attributes of this technology and make point of procedure diagnosis possible. So for people who are not familiar with point of procedure diagnosis, what it means can be for several applications. It could be for needle biopsy. So if there is a suspicion that there might be some tumor growing inside your body, what we can do is we can use needles to extract some of the tissues and look at them using histopathology and microscopy. And another application is intraoperative imaging. So in the case of breast cancer, if there is a tumor within the breast, you can go for this procedure called lumpectomy, which means that the doctors will try to dig out all the tumor tissues and leave the good tissues intact in your body. So what is happening right now is that when we are doing this surgery, in theory, we want to remove as many tissues, tumor tissues as possible. And so in order to make sure this is correct, we evaluate the tissues that is on the periphery of this operation, which is called margins. So in the middle of the surgery, these margins tissues will be sent to the pathology lab. So if they are evaluated and assessed as cancerous, that means there is tumor residue within the body. And because this assessment, it cannot be done in real time, so there is a 30% chance that this will happen and the patient will actually come back for a second time surgery just to remove the residue tumor out. So our thought is that, can we do something about this? Can we use our real-time imaging capacity to build this system in the operating room and then directly acquire images from the margins without any processing within the surgical framework, and then acquire these real-time images and use neural network to make the diagnosis. So because there is a huge contrast between a tumor cells versus normal tissue in our images, as you can see, so the tumor cells have very unique color signatures in terms of the optical signatures, and also they have very unique morphology compared to normal tissues like the collagen fibers you see here. So it is very straightforward for a neural network to classify between cancer and normal tissue. The challenge comes from the availability of the training data for the real life intraoperative settings. So because although we have very wonderful collaborators in the clinics, it is still the opportunity to get into the operating room to acquire tons of this data is very limited. So in order to avoid overfitting, we want to use our benchtop system, which is a much bigger system that's sitting in our lab to help train the neural network to classify between cancer tissue and the normal tissue. So when we directly use the lab-trained neural network on the intraoperative data, the results are as bad as a coin toss. So we could just directly guess, and it could be as bad as that.
Before you go too far, could I ask a quick question?
Yeah.
How does this work during a surgery? So you have the surgeon doing the surgery, and then are they taking a sample? Are they taking? Is there a small device they're taking a picture with, or can you describe that a bit?
Yeah. So really, how this works is that in this surgery, so there are like you have like a few surgeons and then 10 nurses running around. So there is no way we can get close to it. So we actually sit in the corner of the surgical room. And so when the doctors are dissecting the tissue, it's usually piece by piece. And then when they have the margins together, and they would hand us the tissue to us, which we are sitting in the corner of that room, and then we will look at margins. And because this is not FDA approved, so we could not tell doctors what we see. We can just be passive observation. So all the correlations are done later. So that is the procedure. The dream here is that we can get the approval and then use the probe to directly look at the patient, so that you can provide real-time feedback for the surgeons to see if you can dig more tumors here, if you should let it be, so things like that.
And maybe you'll talk about this more later, but I'm curious about some of the other aspects of this. Like, for example, would you need a camera which is keeping track of where the sample was taken from, so that you could provide some of that direction? And also, I'm curious what hospitals you're doing the study in.
Yeah, so we can definitely talk offline, but because the question is actually, that is a very good question. So there is a big problem for the registration. So a lot of the challenge is actually how to correlate our data with the histology data, because everybody sections the tissue in different ways. And yeah, so that is definitely something we can talk about later. Yeah. So here we go. Oh, by the way, I forgot your second question. What is the second question?
Where are you getting this data from? Which hospitals are you working with?
So we were working with the local hospital called Carl at Illinois, which I don't have access anymore. But there are tons of hospitals in Boston. So we very much look forward to collaborate with clinicians in terms of needle biopsy and also pathology. We don't necessarily need to go to the surgical room just for the development technology, but to improve sensitivity, pathology lab is also great enough. When we directly apply to the intraoperative data, the results are really bad, because as you can imagine, when we are in the lab, everything is super ideal. Everything is dark. It's just black, and then you have tons of optic tables to provide you stability. But when you're in the surgical room, first there is a strong floor vibrations, because the surgical room is very busy. You have tons of people running around, and also there is strong ambient light noise, because we need the surgical lights to avoid shadows in the middle of operation. Also, there's limited imaging time, because the last thing we want to do is to interfere the surgical flow. There's also serious issues of image quality, because we started from like a room size system, and put everything inside this tiny box. So there is a lot of image quality issues. It's like we are starting from an iPhone camera and then down to like $100 phone camera. So with these problems, we start to simulate these degradations, because we can characterize what noise and what instabilities there is in the surgical room. So we can randomize this degradation and then train this on top of the benchtop system. And this could be improved a lot after we incorporate these non-ideal lipids in the surgical room. And then for the next step, so this is far away from being good enough. First, we need higher speed because right now we can only take a few sample, so we cannot scan the entire margin because our technology is too slow. It's like 1 second per image, and per image is only like half millimeter by half millimeter. So how do we achieve higher speed while not compromising any accuracy? So this is the idea that is proposed in the Jameel clinic project. We want to do real-time adaptive tumor detection. So in some areas, it is quite obvious that this is tumor, or it is quite obvious this is all lipids. Why are we wasting all these resolution and also different channels to look at something that is already like 100% sure? So one thing we could do is that in addition to have a tumor segmentation mask, we can also have the uncertainty mask, and we can just tell the system to acquire more data when it is very uncertain. And so in order to do this, we need to achieve two technical goals. So in terms of optics, we need to revolutionize how we do optics for diagnosis, because most microscope system we have right now, no matter it's for research or for commercial use, is very ad hoc. We have this laser source, we have this lens, we design everything in a very fixed way, and it can only produce images like this. So my lab is very interested in how to design adaptive imaging system where we can change a laser source, we can change the point spread function, basically like how lens work, and also different color filters at demand. And then the second technical work is how do we fuse this neural network within the hardware streamline to facilitate the real-time feedback. So this is what we are working on now in terms of this project. So in summary, I hope I gave you an overview of what we can do when we combine optics and artificial intelligence, at least in the aspect of improving real-time optical biopsy. And looking forward, I really think there's no better time to work on these problems than now because of just the recent surges in advances in photonics and also data science. A lot of these fundamental problems in the bioengineering can be overcome by leveraging the forces in this. So the first problem we're super interested in is deep in vivo microscopy. One thing I did not mention is that although we said everything is label-free, we did not manipulate the tissue. We could not penetrate through the skin. We have to expose the skin. So in the case of the OR imaging, we can only image as the surgeons dig through the cavity. We cannot, like MRI or CT, see through the whole body. So, so how do we overcome that fundamental limit? And when we overcome that, how can we bring that to the clinical scenario is a very challenging but exciting topic. And on the other aspect, the entire system can cost millions to build, and it definitely can bring down the cost if we streamline this. And also, how can we use the power of computation to bring down the burden of physics and instrumentation? So that's also another active area that my lab is working now, because a lot of the information is actually redundant. So we can use very expensive, very new, very fancy systems to collect the grounded truth, and then find the correlation, like which features are the most important to capture, and then just design hardware that's specifically tuned to that, which will bring down the cost and burden a lot. So those are the problems we're very interested in, and all these rely on a lot of innovations and projects from the perspective of optics, photonics, and algorithms. So with that, I'd like to thank our collaborators in UIUC again, and especially my PhD advisor, Stephen Beaupart. And this is my new lab at MIT. We are enthusiastic about computational biophotonics, and these are all the things we're interested in. With that, I'd like to thank everyone for your attention, and I'm happy to take any questions.
Thank you so much, Sixian. That was intense. You know, I'm still trying to process information here, and I'm sure there's going to be some questions. But just for me to see where we are, because we're talking about visual, right? Here, anything that you are running, this is optics, right? So we're not getting into the, but still we're talking photonics. Okay, I'm trying to understand with different studies that I've been seeing from different groups. Is there any other group that is working on that space here at MIT, Sixian, that you've had?
Yes, so my neighbor, Dr. Jim Fujimoto, who is the advisor of my advisor, he actually is the founding father of this field.
Okay.
So probably used his technology without realizing it. So he and his group invented this technology called optical coherence tomography, maybe 20, 30 years ago, so if you go to hospital now, if you have any, unfortunately, eye diseases, this is what doctors will recommend you to use. So without this technology before, you can only use fundus camera to look at very shallow layers of your eye. But now with this technology, you can look super deep. So you can look through the entire retina and identify eye diseases, diabetes, all diseases. So that's what their lab is famous for. And then their lab is also interested in this area, also using a very similar technology, microscopy, but we have different philosophies. So Dr. Fujimoto is more interested in high speed, but he doesn't care about labels. So they use all kinds of dyes so they are working on instant dyes. So instead of waiting for days, they just want to dip the tissue into dyes and then use nonlinear microscopy to look through this margin very, very quickly. My lab is like more interested in going all the way to label free without the need to go to dyes. I have been talking to him a lot just to see like which direction we should go more to really advance the field forward.
And that actually connects with my next question, and then I promise to shut up now. Connecting what David was saying, do you have explored any local collaboration here with any of the medical institutions?
Yeah, so actually, a lot of people have connected me with clinical collaborators, and I have been procrastinating because the thing is that we do need to build the system to use in the hospital. So right. Our lab is actually more focused on the technology development and then the deployment. So also this is something that I really believe in. So I think the technology right now has a few key limitations that is waiting to be solved. So we can always go two ways. We can always, first deploy it in the clinical world and solve these problems later. And then I guess I'm taking the first approach. I want to solve these technical problems first, and then a talk with clinical collaborators at the same time to make sure that the problems we're solving are indeed the problems they're caring about. Yeah.
Especially if you are looking in the future, maybe to get some kind of FDA policy approved or anything. And okay, that's wise. Okay, so fantastic. Thank you again very much. And I don't know, maybe David, I'm sure he has follow-up questions, or anyone, please.
I definitely do, but I'll let other people go first.
I see a question in the chat.
Yeah, please go ahead.
This is a very interesting question. So as I did my PhD, I actually observed that there is a huge progress in terms of radiation. I remember in the beginning of my PhD, when we did this diagnosis, everything was always as predicted. But I remember at the end of my PhD, when we are looking at surgical samples and when we are looking at pathology report, actually, a lot of surgeons will say there's no tumor at all in the cavity because the radiation was so successful. So I think these two are complementary to each other, because if you do not find any tumors, So that's also good news. And the second thing is that the surgical intervention is not all bad, because when you get the surgical intervention, you actually have access to the tissue inside it. So you actually have the chance to do a more thorough examination. So I do think these go hand in hand with each other. Yeah, so that's my 5 cents.
Thank you.
David?
So I might have missed something in the beginning.
Yeah.
Could, from a technology development perspective, could you make use of samples that are coming to any path lab before they're stained?
Yes. So so that's sort of, are you asking about like the samples? Okay, I think I kind of understand what you're asking. You're saying that there is a gap between when they are dipped in the stains and when they're in the body.
Yeah, so exactly. So I'm trying to get a sense of what the most urgent technology innovations are. Is it sort of creating something that can do it in the body and have sort of the right form factor and cost factor, or is it the label-free aspect of it that you were alluding to earlier?
I think it depends on the application. So we are targeting, my lab is targeting 2 applications specifically. If it's for in vivo imaging, for example, microscopy or needle biopsy. So the needle biopsy is that we don't even need to take the tissue out. We can totally just insert the probe inside of it. And also this probe in real-time detection, then in that case, we care more about label-free and speed. So, and then the third thing is kind of out of control, the FDA. So this is actually approved in Europe and Asia. So, so in Europe and Asia, it's okay to use this technology to look at your skin. So, but for technical limitations, for the real-time interventions, I think label-free, the intrinsic contrast and also the speed is the key. And then the second type of application is more like pathology-wise. Is instead of microscopy or real-time needle biopsy, is more pathology-wise. So so we can totally do something like a alternative pathology lab, right? So so we don't need to do it real-time. We can use instant things. All we need to do is to produce the results instead of taking hours to days. Can we just do this within the surgical flow, right? Because they're easy.
So I understand the use case, but I'm just thinking in terms of sample size, right? So one approach would be get training data by doing a bunch of these endoscopies and just recording the videos or whatnot, or being in the surgery room and getting the samples. A very different approach to get large sample size would be to partner with a major path lab that is getting needle biopsies from patients already, and then before they're, or at some point in the process, I don't know if this makes sense, you can correct me, before it's stained, sort of, get the before pictures. Now you suddenly have millions or tens of millions of these examples, potentially coupled with now the H&E stains that you might get later that you could use from a learning perspective.
That's a great idea. Actually, maybe I should be more actively seeking after this idea in Boston. So we tried to do that in Illinois, but it's a very small town. So we got like 50 patients an entire year. But that is a very good point. And so when we were trying to do that, first is the lack of patients, and then the second part is the co-registration, which you also talked about. So so imaging is fun, but so we have this technology, but we need to correlate with the
exactly. But you could build a device for it. You could put it in the path lab at Brigham Women's and process every sample that comes in.
That's a great idea. So instead of just working on this part, we can kind of streamline the entire process.
And this is just for data gathering, right? And I understand that, like, prospectively, you want to use it in a very different way, but it's to get a huge data set that you can use to sort of build the machine learning aspects of it. Then you can fine tune it for recognizing if there will be data set shift when you start to do it live.
That's a very interesting point, because yeah, that is both for data gathering, and also even if this technology is successful, we still need to do histology later. So this could also serve as kind of like a proof of concept pipeline.
Yeah, exactly.
Yeah.
Sorry, I'm Kumanch Christopher from Memorial Sloan. We have a similar technology called reflective confocal, and then we had the same idea as David, like using ex vivo tissue to simulate the in vivo imaging and then do some machine learning on those. Sometimes the problem can be like the structures in the tissue may change when you excite the tissue because of like it's like you cut it out and then like there's relaxation and everything. And also like at the end of the day, like even if it's a fresh tissue, like some part of it is just like changing its like shape, and then like it's dying at the end. So it may not translate maybe 100%, but like it's doable. Like if you're looking at surgical margins or so, because you're looking at the mirror of the like area anyways, but still there is no one-to-one correlation. As soon as you cut it out, like things start to change, and then hemostasis and all those kind of things start to happen.
That's also an area where, you know, it's exactly that type of shift which the machine learning community has been increasingly doing research on in the last few years, and how to deal with the fact that you don't have an apples to apples setup. One can imagine even trying to like build simulators to try to simulate the live setting with from the
or at least doing pre-training on the data that you suggested, and then instead of using ImageNet or something, like you're using that data, which like 90% or 80% reflects the real conditions, and then you fine tune over the new data that you're collecting from in vivo. With a small amount of data, you will be able to solve the problem. Yeah, so we're also looking into those kind of things for reflective confocal too. Yeah, and then this can be applied here too. The only problem is, as I said, like some needs to be careful about like how the tissue changes, how the structures changes in the data.
Yeah, that's a really good point. A lot of structures we just cannot find it later. It could be destroyed during the staining and processing, and some structures just completely lost, like the lipids, extracellular vesicles. And for the translation, we did go through the same struggle. So we did do a study, which was actually not super good. So I did not show it here. How do we translate from this to H&E? But because we wanted to apples comparison, so the training data was very limited. So in the end, maybe style transfer, maybe like on a neural network that does not require pixel to pixel registration could be the key to solving the problem. Yeah. What is happening in label-free microscopy or this virtual staining is right now is that we're trying to establish a data pipeline, acquisition pipeline that can directly acquire these core-registrated data, which I don't know, which could be even more challenging than maybe doing this from a data science point of view is simpler.
Can I ask a question very quick? So you said you can only do surface imaging, right? Or am I missing something?
Yeah.
Okay, and then how do you think like the depth is a kind of like, I mean, a bottleneck like for your technology? Because at the end of the spectrum, like we have the OCT, which goes like 0.5 millimeter deep, but like they don't have any contrast, only reflective, like reflective kind of contrast. And then we have RCM, which goes to 300 micrometers. Again, we have that kind of a problem. We cannot distinguish in between, like you guys, like some really specific structures. And then you have this beautiful technology and all the contrast, but it only works on the surface. And then there's also competing technologies like Muse or so, which can do the same thing by just using like cameras or so. Like, where do you see like the depth can be solved in this situation, or like do you see depth as a limiting thing in your technology?
Yeah, so Jameel also and I talk about this, like so they started this with OCT, but just like you said, lack of contrast. So every technology always has this pros and cons. And so our technology provides the contrast, but so Muse can only go maybe 10 microns or even less, but it's super cheap. So that is the advantage. And confocal is between, I guess, between Muse and multiphoton, since it's cheaper and the depth is probably half of multiphoton. So for this technology, have to, if I'm honest, if I work on this problem 10 years, I will probably be really happy if we push that limit from 1 millimeter to 2 millimeters, 3 millimeters, which can solve some problems. Like you can use this to look at brain. So instead of stopping at very superficial levels, you can actually look at where the memory layers are, things like that. It's the same with the skin. But in order to really look deeper, I do think a minimally invasive fiber device could be a more practical clinical solution. So so there has been a lot of advances in terms of fiber development. So how do we use like a 50 micron core fiber, basically even smaller than needle, to go into the site of interest? So
and then,
go ahead.
As a follow-up, have you ever thought about integrating another modality into your directly through your objective lens, maybe, so that as you described, you can, let's say, just making it up, but you can do OCT or RCM through the same objective, go deeper with a faster imaging rate. You can just collect larger areas, and as you suggested, only in the areas of suspicion you can collect those your images, SLAM images, in a smaller field of view. So it will make things I guess much faster, but then the problem becomes can you be able to use the same objective lens to do the imaging, or say, have you will you be able to use the same laser to do the imaging at the same time? So those are optical problems, so I guess.
Mm-hmm. So multimodal is definitely one way to go to compromise, or either for compromise or leverage all the beautiful things about different modalities. I think one of the very promising integral approach is ultrasound. So ultrasound is portable, and also there is a lot of interesting interaction phenomena between ultrasound and optics. So Li Hongwang from Caltech, they developed this technology, photoacoustic tomography, that has the combined resolution, optical resolution, and the depth of ultrasound. So I think by combining these two, we can definitely have a much better solution in terms of both resolution and depth. Is it David again? Oh, or is it your hand not?
Yeah, I'm curious about other applications of this outside of the one that you described. So here at the Broad Institute, for example, there's a lot of work on single cell sequencing, and one of the broader questions I understand is to understand the microenvironment around a sample. And I'm curious whether that's something that might be enabled through sort of a combination of your imaging as samples, perhaps. I mean, it would have to be done. I don't know how one would select the cells that one would do. I guess maybe some small biopsies here and there, but just sort of to contextualize the single cell data.
Yeah, so that's another great question. I have been also talking with this professor in Harvard Medical School, Jen Shu. So they did this work. So they are super interested in single cell sequencing with the context of images. So they use Raman. So Raman is one of the label-free modalities I showed today. So they use Raman scattering to collect the chemical signals, and they showed that by correlating the chemical images with the sequencing spatial maps, they could find the correlation. I don't know if they could predict, but they could find correlation between the two. So I think that could be very interesting, how to integrate the vast sea of knowledge in genomics with images. The co-registration would be again a technical challenge. But there is a lot of laser micro dissection things that we can do. So you can shoot laser to burn the tissue off, and then to extract the DNA from that spot size.
Yeah, exactly. Like I'm imagining. I don't know anything. I'm just brainstorming here. I'm imagining going with the endoscopy example,
Right.
where somehow you have a collection mechanism as part of it as well. So like you sort of take, you've got your video, you take small samples, as very, very, very small samples as you go, and you're sort of solving the registration problem at the same time as you know where the samples are coming from.
Yeah, that's a great idea. That's a great idea, because that's right. Because you need to take tissue from very interesting. You cannot take tissue from every area, so it can be kind of like the guidance for genomic sampling. That's super cool. Yeah. Okay, I'm just taking notes.
If
Yeah.
we continue brainstorming, that's getting very exciting. I love this. I love this.
Yeah, I really appreciate the question from David, and also I actually don't get the name of Kibank.
I think he mentioned he's from Sloan Kettering.
Yeah, Kwanch. Yeah, my name is Kwanch. I'm at Sloan Kettering Dermatology Service. Work with Rajat Yaksha. Like Dr. Fujimoto knows our group, I guess. So hopefully we will meet like in one of the SPA conferences or so. Yeah. And I think my colleague Aditi Saur, who's working on tumor microenvironment, also knows like your work. We talked about that a lot, so we know Steve's work, and so.
Oh, that's fantastic. We should also talk more offline. Oh, this is a great opportunity. I actually, because I have been very obsessed with building systems in my lab, I have not think about these problems for quite a while. So this is very stimulating, very interesting. Yeah.
So, Sixian, do you mind typing here your email so people can reach you out? And David, you know him because he's around, but he'll be happy to. Fantastic. And then it's going to be much easier. So unfortunately, we are already on time now. And I think it has been really like an enlightening and exciting discussion today. We have also the recording available as usual for someone that like myself, I think need to review some of the points here. But I think we really enjoyed, and thank you again very much, Sixian, for your flexibility, getting this meeting today, and for the brilliant exposition and the presentation. Thank you so much again.
Thank you so much.
Thank you.
See you.
Thank you.